Okay, let's start. Again, I like to talk too much outside during the break, but I like that. So let's now transition to a little bit of machine learning. Again, I just want to give you a feeling of how it can be applied to combustion. So when I talk about machine learning here, I also like to emphasize that, especially in engineering, mechanical engineering, we need some applied machine learning. Okay, some of us are developing, but I think what really makes a difference in engineering is the ability to apply the stuff to our problems. And so, as you are interested in research, I think the interface between molecular simulations and machine learning, it's a great area for the applications, okay? Traditionally, this machine learning is done a lot in computer science. In Michigan, we have this amazing department, and there are waiting lists to get into classes. But what we also need is a lot of those new generation that can basically embrace both of them. So not only the engineering or the molecular simulation, but also the machine learning, so the applied machine learning. That is not easy. That is, those people who are at the interface is never, they always have a hard it, okay? You need to speak different languages, and has been an adventure for me. So I have a lot of colleagues in computer science, a brilliant mind, but the applications, it's a different area for them than what basically you do theoretically. So it takes a lot of time and to develop something. And that's a good thing, but the point is that we needed to be at this interface here. So when we talk about the molecules and what we had so far, so great, we did this molecular dynamics, but now I have a lot of data and I need to extract some information. So relationships are complex. How strongly two molecules might stick together, or one molecule, for example, transform into another, depends on a lot of details. And I gave you some examples. Those detailed simulations, whether it's the gas phase or the molecular simulation, can be time-consuming and computationally very, very expensive, especially if we need to explore many different molecules, okay, and look at different processes over a longer period. So we need something that is a little bit more efficient, but also that provides some insight. And so to advance this molecular science, we need some way to get into this complex molecular behaviors or database for us. So machine learning can help answer more complex questions. So how strongly will these two molecules interact, or what property will a new material have, or can we predict the evolution of a system? So the power of machine learning is you basically learn from data, okay? And this is an important point, because what is data, okay? And to find, you can find patterns, you can find making prediction, but also you can discover something new when you do this, this type of simulations. So my, something has changed here. We'll go anyway. Definition, basic definition that I wanted to bring with you. So the term machine learning is in 1959, Arthur Samuel, and is the field of study that gives computers the ability to learn without being explicitly programmed for that. And so what he did, he was a program on checkers, basically. And eventually, this program defeated also nearly there was a Connecticut State Checkers Masters. Okay, and this was a game in 1962. So the idea is, I think the real winner here is, as I say here, is the demonstration of learning. So it's not about who won this checker game, but the fact that the computer actually can learn something to do that. So some other definition. People use some of those interchangeably. So when we talk about artificial intelligence, is basically, you can see, is the bigger rectangle here. And it's basically any technique that enables the computers to mimic human behavior. Within this first box, there is what we call machine learning. That is basically the ability to learn without being programmed for that, okay? And then Deep learning is something a little bit different, is extracting patterns from data using neural networks. So they are both part of what they call artificial intelligence, but they do different things. Okay. One other question, and this has been a big discussion, is that why now? What has changed? Because, you know, we saw the beginning, 1952, and then 59, we talked about this, but why now is becoming a big boom? So I will say that there are three main reasons for that. So the data, we have a larger data set that people produce a lot of data with now ability to simulate. Storage, easier to collect also. And then hardware, so the graphic, the GPUs and system have become very highly parallel, if you want. And then software, there is a lot of toolbox, and I could do that. So if you bring together all those three, this is brought us to a time in which it is ripened to look at the machine learning computation, and it's something that now everybody's doing every day. So within these different methods that you can have with machine learning, there is some that is called supervised learning. So what that means is a learning from examples. So imagine a simple example, you have to teach a child to identify apples and orange. So you show them many pictures and tell them, oh, this is an apple or this is an orange. So you label those images, right? So supervised learning is similar. You give the computer many examples, and we already know the right answer. So we show various molecules, in our case for MD, as input, and tell their exact sticking strength. So we took the dimer, we just show you, right? So the right answer or the label. So you say A and B, I did my MD, they really stick together. I did the free energy, good. What is the goal? The machine learns to find the relationship between the inputs and the right answer. So they can predict a new unseen input. This is important. So unseen input. So you train your machine learning on some of the data, but can I predict? This is the challenge. This is something that you're really interested here. And so the focus for predicting molecular property, we use those supervised learning at this point. And so let me give you some examples. Supervised learning, it's basically learning a function that maps an input to an output. So I have my A and I have your B. And you decide which one they are, right? So if you have, for example, input A, email, then you want to know out B, is this a spam or not? Then what you're doing is basically the application is a spam filtering. If you have as input an audio and you want a text transcript, then you're doing speech recognition. So your A is here, your B is there. Or image of a phone, if there is a defect, you're doing a visual inspection. Or image or a radar sensor, the position of other cars is your output, then you're doing a self-driving car. So again, you have your input and you have your output, and you decide which one they are. So in this case, simple A and B. But what is data? That's one of the main issues, because there are algorithms, but whatever you put in, garbage out, right? So data is important. So what are we talking when we say data? Well, data could be an image also. It could be a number from MD, but it can be an image. So you have an image here, and you can say, label, this is a cat, okay? Or here, this is not a cat, this is a cat, this is not a cat. What if you're interested in buying a house, for example? So now you start saying, okay, I see that this is the size of the house here. This is the number of bedrooms, and this is the price. So you decide your own A and your own B. You can say, I want a size, this is the size of the house, and this is the price, so this is your B. Or you can say, my A is the size of the house, but also the number of bedrooms, okay? And so you are making this a little bit more complicated than that. So you decide, it's up to you to choose A and B. So with the supervised learning, you primarily have two main problems. The first one is classification, and the second one is regression. Classification is, like I showed you, is basically deals with the predicting categories or label. Spam, not spam. Cat or a dog, or not a cat. Regression is instead interested in predicting continuous numerical values. Okay, so not just cat, no cat. And that's what I need. I need eventually to come up with free energy, so I need a number. So we are going with regression at this point. And so what is regression? Within supervised learning, regression is about predicting a continuous number. And so predicting the temperature tomorrow, predicting a person's height based on their age, or a molecular property, like how strong two molecules will stick together, those are all examples, okay? So imagine that you have data point. We have a lot of data, scattering data. So molecular features or the sticking coefficient that we look with molecular dynamics. What regression tries to do is to find a trend or a relationship in this data. And so the best fit line, if you want to simplify the project, I want to give you a simple idea. And so you might imagine like a straight line through your data point, okay? So this line, once you have the line, that helps you to predict what comes next, to estimate. So this is an example. Let's assume that you have a lot of points, they're scattered. The simplest thing that you can do, you find a line that best fits all the data, right? And then this helps also to predict, because you say, okay, if I am here, maybe this is the number, my point will stay here, right? This is the prediction. But data also can be very messy, so you need to pay attention to what kind of data you're looking at. So garbage in, garbage out is what I was mentioning. And so the data problems, you have incorrect labels. You can have missing values in the data. You can have multiple types of data that you're dealing, so not only numbers, but it could be image. So in a flame, I have a spectrum, a mass spectrum. I can have an AFM image. Then I have an MD simulation is a number. All this gets into the problem. And so if you see here, this is an example, again, the size of the house and then the number of bedrooms. So you can see that this starts being a problem. This is unknown, right? But this one, also the price, that will be great, 0.001 here, okay? So data, you have to be careful when you look at the data, what you have, so, and make sure that they make sense also. Okay, so machine learning tends to work very well when it's learning a simple concept, okay? Whatever you can somehow think or give an answer quickly, that is a good problem for machine learning. There is a lot of data available too. So usually they say, we need a lot of data. How much data do you have? When people ask, can we do some machine learning on this? I say, how much data you have? But instead, it tends to work very poorly when you learn complex things, complex process. Remember that I told you that if you have molecular dynamics and now the binding between two molecules depends on oxygen, mass, and there is intertwined, this is going to be a complex process. So, and if you have a small amount of data also, and then it's asked to perform on new types of data. So that's also the two major limitations that is now seen in this training. So this is an image of pneumonia in the lungs. And so the thing is that if you have 10,000 labeled images, okay, then the code can predict if this person has pneumonia or not. But it cannot diagnose pneumonia from 10 images of a textbook, okay? This is another example. You have to be careful about the data. So you see here the lungs, are, if you want to straight up. But if the image, for example, is by any chance tilted a bit, or if there is any kind of different pattern, corrugation, you scratch the images in some ways, then it becomes a problem. Those are simple. So he recognized this, but it doesn't recognize now this one down here. Okay. So this is a piece of the data. The last thing that I want to show as a brief introduction is that what is data science? Data science, so machine learning gives you computers the ability to learn, as I was mentioning. Data science is extracting knowledge from data, okay? So what you see here is that maybe, okay, this is a house, you know, the number of bedrooms, maybe I can go to 1,000, I have now 3 bedrooms, and that could be a better deal, for example, okay? So you learn how those numbers are what are the trends between those numbers somehow. So extracting some new knowledge. Okay, so let's, with this a simple approach, let's see what we have done for the growth of pHs. Now, the problem here is that there is a lot of features. We've been talking for 2 days about this, and they have, there are many characteristics. So features means characteristics for me, kind of said. And so there is a size, there is a shape, there is flexibility, they might contain oxygen and so on. And the problem is not all of them are important, okay? So an example, you want to predict the house price that I told you before in the tables. This is the size of the house is important, but the color of the mailbox is not, right? But that could be all part of the data that you get. And so the model can get confused. This is a noise, is not important detail in this moment. So it becomes, it might become too specific to the training data, okay? And so he fails to perform, and this is called overfitting, as you want. So going back to one of the questions that Patrizia had before is, how long it takes. So if I am doing one free energy for, depending a little bit on the temperature also, but on average, I will say maybe 3 days for that. And so what you see here is the number of unique pHs that you have in the system increases. And as you increase the number, now the dimer that you can form, the pairs of dimers, goes up. Okay, so 80, I have almost 4,000. So, and if you want to be very accurate, again, this is a lot of information. In the flame, you have between 2 and 6 million of species. So this is not doable with MD. So machine learning maybe can learn from the simulation and predict the dimerization for any pairs of pHs. Okay, that will be the goal of the interest for this study. So overall, the idea is that I'm taking molecular dynamics data set that I showed you before, in which I have different pairs of molecules getting together. I can compute the barrier here, so the hump to go over to become a dimer. And then what we do, we translate this molecular simulation into some kind of a machine learning algorithm that can give you, what am I interested in? I'm interested in predicting the barrier. Okay? So I don't want to run a lot of MD. I'm tired of that. I just want a way to get this number for any pHs that I want. So we need to find the right balance, because this I'm showing you 3 different scenarios. So if you have blue are your data, now what you see here is that if you go with a model that is too simple, you can have something that looks like this. So it's too simple, does not capture the behavior of the data, right? And so this is called underfit. If you go to high, what you have is very complicated. So look at this red curve that you have here. It fits all the existing points, but generalizes very poorly to new points. So this is what they call overfit. What you do is a balance. So you want a good fit that there's a proper complexity, because you need to retain some of the features that I learned, but also can capture the data and generalize. So for those problems, we have been using lasso. This is a very cool approach, and the reason why it is interesting is because it can give you some information on the features as well. So lasso lists absolute shrinkage and selection operator. Lasso is easier. And is a regression. So the idea is exactly what I showed you to. They solve too many features problem. So what it does, this is the objective function that you want to minimize, and includes basically the goodness of the fit here that goes, so penalize poorly fit models. And here is a kind of a term, so this is the truth, and this is how far from the truth you are, but then there is also some kind of a regularization term here that you have, and that penalize overly complex models. So what you're going to do is to look at 2 things. You don't want it too complex, something that looks like this, and you want to do some goodness of fit. So you want to be sure that you are not in this scenario, and you are not in this scenario. Okay? So he combines those 2 strengths and basically try to minimize those two different extremes, if you want. So what it does is basically is like a smart filter, if you want. And so he identifies and picks out the most important and relevant features that influence that L function that I showed you before. And then he ignores the noisy ones. And so makes the model more reliable, especially when it encounters new molecules. So the idea is that let's stay away from those extremes. Let's try to minimize those two things and try to get in the middle when you have a good fit. So for the house examples, for example, Lasso will automatically realize that the house size is crucial. And you don't need to worry about the mailbox, okay? So that's the goal here. I want to make sure that for my molecular simulation, I can filter out the important property that drive the dimerization and not everything else that you can compute. So what do you do before running? Lasso is the mathematical equations, but you need the data. So what you do is, let's talk for a second about training data and test data. This is very important. When you work on machine learning, you want to distinguish that. Training data is like the material that you study, so data that you feed to your machine learning and is used for the study. So the model, Lasso, studies those data and looks for patterns, the relationship and the trend, okay? And then he adjusts that internal settings and the rules based on what he learns, right? The L was auto. So think about textbook, lectures, practice problem to learn the subject. The test data is the final exam. So that is a smaller amount of data that you have, and the model never sees this data until after he has finished learning. So how it's used, once the model is trained, so you go this training data, we unleash those test data. We then compare his prediction on this new data and to see if he gets the correct data. So in general, just to give you an idea, one technique is that let's say that you have 100 points, okay? You split those 100 points in beans. And you say, my training, I'm going to give you 80 points, for example, and you reserve the 20 left as a test. So your system has never seen those 20. You train on 80 points, and then you see if you can predict the 20 missing, okay? So you need to, he never sees those extra 20. Why this is important? Because it tells how well the model will perform in a real application that has never seen it before. If a model performs great on the training data, but poorly on the test data, it means that he memorized the training data too closely. So it's an overfitting and cannot apply to new situation. That's not what we want. If he performs poorly on both, so the testing and the training, he has learned enough. So he's underfitting, okay? So let's take a look at how it works here. Again, you've been with me, you know that we have this pool, a lot of molecules, lot pools. So what we did was to consider a set of these pHs in different sizes, shape, oxygen content, and functional group. I also included this, the rotable bond, because it's very important, as I mentioned to you. And then we look at simulation that we did, and I showed you the result before at these 3 temperatures, and the combination of homo and hetero dimers. What I mean here is that homo, 2 molecules, A and A, hetero is A and B. So I can have this monster here reacting with something here. Okay, that's an hetero. Because of course in a flame you don't have 2 things only, but you have a variety. So what happens here? The simulations that we tested with the molecular dynamics, they scale linearly with the number of temperatures and combinatorially with the number of pHs. I showed you the pH before. Here, if I have a pHs in the data set and the number of simulation that I need, if I go from 1 temperature to 3 temperature, you can see that, let's say for 1,000 of those, in blue, I have, what is this, 10 millions, whatever? No, 1 million. Here you go to 100 millions, okay? So not doable. I need, again, I want to be able to run, for example, I would love to run my MD in, let's say, 1, 2, 3 simulation, 1, 2, and then predict for another temperature, okay? So what I'm asking is a way to extract features that are important for the formation of dimers, first one. The second thing is that can you predict the dimerization, but also can you predict new temperatures that you have not seen before? So this is what I would like. And so let's see the question. Again, I'm not interested in seeing how well a model can fit known data. I'm interested in saying how we can predict new data. And so for the pHs, for the case, given the free energy of some pairs of pHs, can the model predict the free energy of new pairs that has not seen before? And what are the most important features of the molecule that drive those values of energy? So here are some of the results, and I think this is a cool feature, a cool figure. I put in the parentheses because the problem was that we have a lot of pHs, right? So look at here, how can I cluster them on a graph to show? Because it's not only the 6-member rings these are fully condensed pHs, right? But this guy is an aliphatic chain. This other is an oxygen. This is a bond. So how do I cluster in a graph? So first of all, let me show you the result. This is from Lasso. So we minimized that function that I showed you. We did some training, and we did the test. So this is specifically we trained on what they call fourfold, and we left one bin of the data out. So then we did the validation with that. So what you see here is a graph that shows this is the predicted free energy, and this is the MD free energy. If you are on the line, the agreement is pretty good, okay? So they have the same number, basically. And so what you can see here is that we color those kind of the sphere, one single color or 2 colors. That's the reason why I think it's cool. Because some molecule can be condensed hydrocarbons, like for example the blue, but they can have also some oxygen attached, right? And so when you have like a sphere that is blue and red, it means that it has 6 condensed rings, but it also is oxygen. So what you see, first of all, is that, and the same with the green aliphatic chains. This was a great agreement. So what we managed to achieve with Lasso is the ability to predict the free energy for pairs of pHs that they've not seen before for the system. This is the mean absolute error or the root mean square error. Those are different ways to distinguish the agreement between the model. But this is a very good number. So it works pretty well. Of course, there are outliers. And then you start wondering why there are outliers here. And so this is not a specific group. So we investigated those ones, not related to or molecule, but are heterodimers. So meaning some of the simulation between molecules that were different. And one of the problems is that maybe in the training data that I gave you, there was not enough information on heterodimers to keep moving, okay? So, of course, you can refine that. You can do a better agreement. Maybe you need to add more data in the training, more MD. But with this amount of data, with this amount of data, we managed to get a great agreement for this system. So this is an important point. Okay, so Lasso, regression, this approach of machine learning can be used, and you need to be careful about also the data that you have and what type of data. This is all good, but this is a black box right now. And the question was, can I interpret those? Remember, we have that expression of L in which you minimize the 2 extremes, and there are also a dependence on the features. So what Lasso does is basically, this is a complicated image, but let me go slowly. Lasso, first of all, how does it perform with other methods? What I show here is the error. So if you are a bit here, it means you make a bigger error. Error is the discrepancy between truth and your machine learning, okay? So if I am at 21.3, this is my error. Now, this work, the one I just showed you, Lasso with the chemical descriptor you basically outperform method when it talks about free energy. So this work showed that those are the two types of errors that you can have. Gives you this 6.4 at 10.2, whatever is the error, okay? Now, if you do something about reduced mass, if you try to predict the dimerization of pH using some only mu that I showed you before, so the reduced mass between the two, for the same set of pHs, you get, reduced mass, this type of error. So 10.2, much higher, and 14.3. If you use, this is the surface area of pH. So remember the steric hindrance was an issue. So if you have a molecule and you start to define a surface that somehow is a kind of a border, so when another molecule cannot get in, just to have some idea of the collision, then this is the error that you get. And forget about the series. If you do just a naive, you do an average of the number. So your MD gives you 100 points. I'm just giving an average. Then this is almost 18%. Okay. So lasso, that does not take a lot. It's a great way to predict the free energy of those pHs. So can we interpret? Well, Lasso select set of features used to make the prediction. So he realized that with that expression that I gave you before, they can see which one of the features is playing a bigger role in terms of the discrepancy with the truth. And so across a small variation to training data and sets that are considered here, so the feature capture size, shape, oxygen, and aliphatic chain. So those are the four main features that basically Lasso was able to extract from the whole pool of MD simulation that we had before. And so in terms of shape, if you have a long, narrow pHs, these are definitely less stable for a dimer. If you have an aliphatic chains, they stabilize, okay? And why they stabilize that? This is one of the questions I got during the break. So if you have, just let me ask you this, and you should know because I told you in day one also. So if you have this molecule, do you see that there is this aliphatic chain here? They are impacting each other with velocity. Velocity means energy. If they are just, let's say that you are just aromatic, very rigid, okay? What they do, they hit each other, they bounce back, right? If you have a way to dissipate energy, then the two are impacting, but now the energy that they have, he goes also in rotation of this guy, right? Remember, this aliphatic chain, the energy goes rotation, goes in vibration. This kind of hand will start rotating. So the molecule will slow down, and then they have a bigger chance to stay together, okay? So the reason why aliphatic chain plays a role is because they can dissipate some of the energy that they receive upon collision. Another important part is the oxygen is playing a role. And actually, the oxygen is destabilizing the dimer. So he plays a role because he doesn't allow the dimerization. So I'm not showing all positive. I'm showing also what causes the opposite process. And of course, the size, the bigger are more stable. So those are the main four features that we identify with this study. So first of all, why this is all important? Because now, if I have pHs from the gas phase, and again, I want to move into a model, an empirical model, what I do, I'll take my molecules, I compute for those. I can say that the rate of dimerization is a function of the size, is a function of the shape, and a function of aliphatic chain. I do need to define them as a numbers or some kind of empirical formula, but I know which one they are. So it can be easier for me to find an expression for that rate of collision. Can I extend? Remember that my point was that I don't want to run so many simulations. So can we extend the lasso to different temperatures? So we trained only on 1,000 Kelvin. And then the question was, can you reproduce 500 Kelvin, but also 1680 Kelvin? So outside the boundary of the training data. And what you see here is that actually the diamond is 500 and the circle is 1680. And so this approach does a great job. Again, you have the predicted free energy versus the MD, the number the MD will give you. And you see that it does a great job. The model predicts the homodimerization at those two temperatures. And this is, again, the error that is 4.1 kilojoules per mole. So for this case, for this scenario, our machine learning has worked very, very well and is actually a way to extend those molecular dynamics and actually extract insight. Again, not just a black box that can help me to speed up the calculation, but also understand what's happening here. So the takeaway, from this one is that molecular dynamics tells us something about how the property influences the free energy or the physical behavior. So the whole variety of property. Machine learning can extend this information to different pHs and provide also some interpretation of the reason. So what are the chemistry, the physics that is driving this? So identify, the main positive is that has been able to identify key molecular characteristics for those interactions. Okay. And just to give you, this is more, I already said this, but this is the last part. So we can predict the dimerization, so how two molecules get together, and then from the free energy, get to the rates. Now, why this is important? Many people, there are different mechanisms. Once you get to this gas phase, people propose different methods. They get together physically, they start forming a dimer, and then eventually maybe they can make a covalent bond, right? And then they can grow with other chemistry. This type of approach, I am not discarding an hypothesis again. The idea is that, first of all, you need to get together. To form a bond, you need to get together. So this is the first step. This tells you the rate by which two molecules get together. Then they can also form a bond and be chemically bound to each other. But they need to get together. And so this tells you what is the rate for those two to get together, to be close enough. So so what we can do now is we can have a model that basically says that the aggregation of pHs is a function of 4 or 5 molecular properties that we're interested in. Okay, so something has changed here, and I don't see what's happening next, so I'll go back. Any questions about any of this so far? Yes?
So for the slide for the reduced mass.
Yes.
And then when you use the reduced mass, the MAE increase. And when the MAE increase, that means the prediction kind of become worse, right?
The prediction becomes worse, right? Yes, yes, correct. So the point is that, so let me go back here. So here, reduce mass here. So what you're saying is that there is a bigger error. Yeah. So the discrepancy between the truth that could be MD and using a simple model that is based on the reduced mass only is bigger. So because reduced mass is only part of the story. There is also, there is the shape and there is the aliphatic chain, for example, that play a role. And so if you pull all of them together, you jump to what I show you, in which gives you a smaller error. Okay? So only one reduced mass gives you a higher error. Okay. Also, you don't predict. You know, if you want to extrapolate to higher temperature, this is not going to help you. So I don't know. Yeah. Maybe that. And also, this is 2 different errors we didn't need, but that's the figures. Any other questions about this? Yes?
Do you know how the uncertainty in what you're feeding into the model affects the uncertainty out?
Yeah, that's a great question. So yes, there is some kind of error that we follow through the whole simulations, but this speaks also to the amount of data that you have. So for example, I don't know if I showed you here, before. I also have an error here. So when I say this is the error that I get with the machine learning, for example, prediction, this is, I compute a different error, but also I keep track of the error that I have in the simulation with MD. So you are absolutely right. There is an error that propagates. So you need to take into account that. And so when I give you an estimate, I also tell you what is the original error in the MD simulations that I was running with. Yeah, absolutely. Yes?
I was just curious, again, like this, the heterodimers here, like you've sort of shown like the heterodimers don't agree with the main data, and even like the next plot, like you've said the homodimers sort of agree with this plot. Like, is there like a characteristic difference between the heterodimers and the homodimers, and like in like a real flame, what like dominates each other?
Yeah. So let's see the book. This is a good question. All great questions. Let me go back to the pictures here. So the problem is that again, the training data is what the computer is using to learn. Okay. So if I don't have a lot of data about some hetero combination, he has not seen them before and doesn't know how they behave, so he's not learning. The reason why sometimes is more complicated from a physical point of view is that if you have something that has a huge mass, and then you have something that is tiny, like L here, then the collision is dominated by this guy. And so these are some of the phenomena that are not included if you don't have a complete training on hetero molecules. Okay, so it's just that basically he has never seen this case before many times. And the physics, it's not the same that you will have if you have L with, I don't know, this one that is much smaller. So there are other factors that now play a role, and since he's not learning on that, they cannot give you that. So because the good things, if you want the good things, is that this one were only, the outliers were only on hetero collision. So we do know that we need to add more data in the training if we want better capture those ones. So there is a physics that has not had an opportunity to see. That's what it is. Yeah. Any other questions? You have a question? Yes?
Do you see different priorities in terms of like what is most important for minimizing error when you're looking at the different like types of molecules or sizes or anything like that?
In terms of error?
Yeah.
What I see in terms, I don't see specifically for the type of molecules that the error is more about, maybe let's get a step back for a second. You have a great question here. So the idea is that what I need, maybe I didn't have enough of the chemistry, for example, with oxygen in the hetero atoms. So when you do those kind of, so remember that these ones are positive, right? And this one is negative. So maybe what I'd see from a chemical perspective in the error is that I didn't have a lot of hetero, but also I didn't have a lot of done with the oxygen. That seemed the negative. So I can tell you more about the chemistry, not specifically the error number, but I think this one was the one that was missing the most. Hetero and with the oxygen for the negative. So you also want to have a negative and positive when you train. So that's also a great question. Did I answer your question? Any other question? Okay, so let's jump for now to something a little bit different, and then we'll take a break. Okay, so I want to, hopefully, I didn't have a chance to fix the slide, so you bear with me here for a second. But I'm always interested in this nanoscale interaction. So it could be nanoparticles. It could be also, if I look here, it could be a nanoparticle and a protein. This is just examples that I want to show you. It's the same concept. It's a nanoscale interaction that you're looking at. And so you can think as application something about some biological function in which you have a protein and you want to see how it interacts with another protein. You can have a design of nanoparticle. So remember, you need to get excited about this field because it's not only Soot. It can be designing of materials. And so this is an example in which you can use some molecular dynamics that I just showed you and see how some new materials can interact with the proteins, for example. Or I'm interested in this nanoparticle toxicity. You remember I told you all the problems related to the health effect. You produce those pHs, you breathe them, and that can cause trouble. So let's talk for the first topic I want to talk briefly is what they call risk assessment. You remember that I said one of the reasons why you're interested in this is because they can cause a health effect. And I show you some image on day one in which they had some experiment in which people People and rats, they were fed with either nanoparticle or big pHs, and they will see them translocating, meaning crossing membranes, crossing organs in the system. So I'll show you that. But when people, you talk about, you have a molecule, and then you want to say, is this dangerous or not? I have a new pH that now Synapse has produced. So think about SAF. You are now switching to a new fuel, SAF. But what are the emissions now? Maybe you don't have Soot, but maybe you have other structures. You have big pHs that you have not seen before. You have a new nanoparticle that is forming. Okay? Those are, as you move into something new, you can have new prediction and new molecules. And this is the reason why I think Synapse helps, because he doesn't tell you again what to expect, he produces it for you. So let's say that Synapse produces a new pHs with a new fuel. Now, can we say something about the risk of this for humans? So usually risk is related to two things, the hazard and the exposure. And so the hazard is just how much damage this molecule does to the body, but then also exposure. So too often this exposure part is neglected. And what people do when they talk about those problems, it just go back to some kind of representative nanoparticle. And nanotubes has been a big one. But the real truth is that who cares? I mean, not who cares, sorry. What I want to say is that what is the exposure level of carbon nanotubes? Okay, it's negligible. So yes, you can correlate, you can infer from the result from nanotubes what could happen if you breathe the pHs, but this is not, it's a leap that you're taking. So we have now a way that, as some approximation, in which you can produce pHs, we validate the existing experiments, and we can also build a nanoparticle. So can we think about something different? And so what I've been doing, and this is, we take, this is a little bit of biology, but not much, but I want to show you how you can extend the tools and how you can relate to combustion as well. So first of all, those are different pHs. They were somehow, this is a more Fullerene, those are macro pHs, but that can be produced, right? And so the question is, will they go through biological cells? So if you inhale them, will they cross the cell in your body? Will get to the lung? Will translocate? So the problem is that here, we all deal with the cells in the body. And so if I want to know if the molecule is moving, I need to cross cells, right? And there are many different cells in the body, but they all have, some three parts that are common: the membrane, the nucleus, and the cytoplasm. And so what you care about is the cell membrane. Your pH is here, it needs to cross the membrane to get in. Once it gets in, it can cause a lot of damage to the nucleus and other parts of the cell functioning, the protein, the signaling, all the stuff. But he needs to cross. Okay, so for me, that I need to do a molecular dynamics, this is huge, so I cannot do that. What we do is basically blow up this section, and we look at the membrane, the cell membrane, and then the interaction with the pHs. So what is a membrane? The cellular membrane is this, and again, it's chemistry. So it's carbon, hydrogen, maybe oxygen, and some phosphorus here. So this is a lipid bilayer, is the membrane. And the reason why I want to emphasize this is because you have an upper part that's called head, and then you have tails. Tails is carbon and hydrogen. The top has some oxygen. So this is hydrophilic. It likes to be in water. Our body is made of water. Right, and so the tails instead are hydrophobic. They don't like oxygen. This makes a difference when your pH is an oxygen or not. So that's the reason why I'm pausing for a second here. So your membrane is going to look like this, lipid bilayer. And this is not relatively important, but basically the idea is that the tails can be different. You can have a single bond, you can have a double bond, and that changes the stiffness. And think about this. If some molecules are diffusing through the membrane, the presence of stiff bond or something that is very flexible will change this, okay? So, So this is how a simulation looks like. So we did simulations of, let me show you a second. This is a lipid bilayer. So the pictures, this is water. water at the bottom and at the top, lipid bilayer. Then you see all this head. Those are the legs, the tails that I showed you before. And you can see some of this orangey stuff. This is cholesterol, because you know that we have a lot of cholesterol in the cells. So we need to take into account that one too. Now, there was a huge simulation, and our goal was to understand, given a carbonaceous material, in this case it was a fullerant, how does, first, if it translocate, and how it does that. And so the other question is, can we develop a model that allows to predict any structures coming out? Will they go in or not? So this is a fully atomistic simulation that we did, because you need all the information. You need the hydrogen. You are in water. There is the hydrogen, bonding is an important part. It contributes to the entropy also, the system. So I couldn't do any approximation. I didn't do any coarse graining. This is all fully atomistics. And so when you change the cells in the body, you will have a different relative concentration of those species, okay? So you can have the mitochondria, you can have the Golgi, you have different cells in the body. And what really means for my simulation is that the relative concentration of cholesterol and those lipid tails will change. So what did we do first? Okay, this is a simple simulation. We took, and again, this is the lipid bilayer. I'm just giving you a different color here. Again, same things, maybe with the head, the legs, and the blue is the cholesterol. Then we took 4 molecules or microstructures. One is the fullerant, and you are very used to that. Only carbon, C60. Then we said, why don't we think about an open fullerant? And the reason why I wanted to do that is because I wanted to assess, these two have the same mass, okay? So mass, I'm keeping that constant, so I want to understand if maybe opening the fullerants will create some problems. So the exposed area will change in this case. Then we did a couple of nanoparticle or macrostructure that we got from SNAPs. And the only difference were that this one is hydrophobic, carbon and hydrogen, and this one has some oxygen attached. Okay, so according to the idea of the effect of nanoparticles, you just have maybe nanotubes or a fullerant that usually is used, so they will behave the same, right? So we did a molecular dynamic, and we wanted to assess, will those 4 molecules behave the same, yes or not, right? And so again, carbon, 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 hydrogen, oxygen. So here are the results. So this is just the example of simulations that we do. We look at how a molecule from outside gets into the lipid bilayer. Again, this is the inside of the cell. This is the outside of the cell. The molecule is coming in. So this is the simulation that you do. And we look at free energy, because now you know a lot about free energy. So this is 0 is the center of the membrane, of the lipid bilayer. So let me go back here. So this is 0. This is 3, and this is outside. So what I'm looking at the free energy of this guy from this position going all the way into the membrane. So outside into the membrane. So the results show that looking at the free energy in terms of distance, first of all, the black is the C60. So it's what the C60 will do if he gets close to the membrane. Then I'm going to compare with the open C60, the green one, okay? And what you see basically down here is that already I'm keeping the mass constant, and I'm changing the exposed area that I have of the structures. Well, it gives you a completely different free energy here. If I have a combustion, now I have a carbon and hydrogen here molecule that is out, then I have the blue line. And if I have something with oxygen, like an hydrophilic, polycyclic, big macrostructure, then I have the red. And the reason why this guy is here is that he loves the heads of the lipids because they are hydrophilic, I told you before. So he loves to be here, where the yellow is. Then when you put him in hydrophobic, he doesn't like that. And so you can see that as soon as he gets close to the membrane, he will jump in, even faster than the others because they are hydrophilic, both of them. But then he will go in, but he really doesn't like it much, okay? So the bottom line is that there is first of all my molecules will get into the membrane. That's number one. You see, this is 0 and this is negative energy, so it spontaneously will get into the membrane. And second, the mass is not discriminant here because I have the green and the black, but the other ones instead also, they have a different energy. So this is interesting, but again, I need to go up in a scale a little bit. And so I can keep running simulations for all of them, but can I predict? Can I do that? So here we do something a little bit different, and this is more the statistical analysis of the result. So I have those simulations in which I always see that I have a stage 1, a stage 2, in which the molecule will get close to the membrane, and then eventually flips here. So here, and then in this case goes in. In other cases, it gets close to the membrane and then it goes away. So what we were able to, we did a lot of simulations. So what we were able to understand here is that the position of this molecule, and actually this was a graphene quantum dots. That is one of the material that is also used for a lot of biomedical application because it's fluorescent, has optical property, but it's a pH, basically, if you see here, similar to a pH. What happened is that we were able to correlate the propensity to go from stage 2 to stage 3 with the formation of some local low-density area on top of the membrane. So the membrane is basically in constant movement. The lipids move in the membrane. And so what we were able, through MD, to correlate the chance of this guy to get in with the formation of pores. So we had to do a lot of work as a great student for that, because one thing that you here need to do is to transform this. You have to read this image, basically, and define what is the open area. So that was a little bit of a challenge. But what we came up was using a lot of MD simulation with this model. So this model, or we call a low-density area model, but what it tells you is basically the probability of any structures, so not carbonation, not particle, to get into the system. So you have a probability, of course, to get close to the membrane. And then if you have a high density that basically is closed, there is no hole, you get a bounce back, you get repelled. In case you have a low density area, you have a probability to go in, okay? And so we did some different analysis and we were able to describe this process with only 4 parameters. And they are decoupled. So this is very interesting because the first one is the probability of finding a low-density area. How do you know that? It's a parameter. But once you run your MD simulations, you have a library of those membrane. So you know what this number is. This is the chance of going into the lipid side, and this is related to the partition of the lipid water for a molecule. And then you have the tau, that is the time for the drag, related to the diameter of the drag. So it's something that you know, because if you have a molecule or a drag, you can compute the diameter. And then the last one is the lifetime of the low density area, and this is also related to the MD. So those 2 parameters are related to MD, and you form some libraries. This you get from the particle that is coming in, and this is the octane, the lipid, the partition in lipid water that is you can compute. So all this to tell you that now we have a way to compute it. From MD simulation, I can compute the time of entry of any structures, whether it's a pH or a nanoparticle, into different membranes. And so, again, I need information about the membrane, and I can do with the molecular dynamics, and then I need to know what's coming in for the system. But, of course, it's always like that. You can model whatever you want, so you always need to work with experimental people. So we went to a colleague of mine, Professor Liu at the University of Michigan, and what he did, we did some experiments. Because now I have this model, but how can I validate? And it was not easy. So what he did is do some experiment in what this is vesicle. Vesicles are spheres of lipids. So they have a membrane, okay? And then you have the nanoparticle, whatever it is, inside the sphere. So he built spheres, this is lipid spheres, so vesicles, with different concentration of, you remember the tails and the cholesterol, so phospholipid and cholesterol, to make sure that we can represent a different membrane. And then he put in some of those nanoparticles or pHs that you can also buy, some of those are standardized. And so what he did was the photoluminescence image of those lipid vesicles, and he measured the leakage. So he measured the time for this guy to leave the vesicles, okay? And so that will be equivalent to what I showed you before in terms of time that we can compute with our model. So we did that, and here are some of the results that we got. So this is the comparison, a different membrane between the cholesterol and the phospholipids, and the agreement between the model and the experiments that we have. So pretty good job. And then one more comparison, because in addition to experiment, you can also compare the permeation of different particles using full atomistic MD. Okay, they can give you. So this shows the prediction of this LDA model, so the blue cross with different MD simulations. So with the agreement is very good. Okay, so let's talk a little bit more about this, and then I'll stop for a break. Cellular membrane. Now, I just show you one membrane, right? But you can do more than that. And the reason why I have this table here is that you can change. It's not easy to build a membrane, but in principle, you can change the concentration of those species in the membrane. That will be here, right? And the cholesterol. And as you move in different cells of the body, those relative percentage will change. Now, who cares? The reason why you care is because say that, for example, you are breathing a pHs. Maybe he crosses the first membrane, but then is not able to go more inside in the Golgi and the mitochondrial that they are inside the cells, okay? Or, so you can have an idea where he gets stuck. Or if you want to be thinking about a new design of a drug, and I know that is a little bit of a stretch here, but drugs are based, they need to reach the target, okay? You take a pill, you take something, it needs to go inside the body. So when you predict the translocation of those drugs in one cell, you need to make sure that it reaches your target. And your target might be maybe the Golgi that is inside. So you need to be able to cross the plasma membrane, other membranes before you get to your target. And so your drug needs to be tested on those membranes as well. You cannot just test on the plasma membrane, the one outside, okay? So here I have a little bit of more, this is human, this is real membrane, so in which you have a different concentration of those species. And we did simulation, so we ran MD. They look like this, again, the membrane, and we checked all this. So, and as since we'll show some result, you will see that the fact that you have either a different chemistry in this is the lipid, in the tail. So if you have a saturated or unsaturated, it will play a role. Because again, a double bond here will make this a little bit stiff, or it will give you some space also for a molecule to get stuck. Okay, so those properties play a role when you want to, you are extending to other systems. So, question. Now I have a lot of, we saw a lot of pHs, right? A lot of hydrocarbons. So I took two extreme. I thought a dodecane, a fuel, if you want. And then I took these fully condensed pHs. That is actually, this is a graphene quantum dot. And the question is, how does it change? Whether you're breathing this guy or this guy will change. Will they cross the membrane in a different way? And then I compared with the vancomycin, that is basically an antibiotic. So with antibiotics, you take it, and they know that they go to the target, they are fast, they act very fast. So let's take a look. So what I did, and it's a lot of color, but stay with me here, is this is what I'm showing you is the time of entry. So if you have a short time, it means very fast you get into the membrane. I look at those three structures coming in, and then I add a membrane with different percentage of those lipids. The reason to do all this study was to understand the role of the double bond, the unsaturation, and the different concentration, so that we did. But what I want you to pay attention to is this part here in red. Those are the real membrane from the cellular, the mammalian cells. So while those are made up proportions, those are simulating the real ones. And so what you see here is that no matter what membrane you are crossing in this way, so it could be, this was from the rats were the membrane, basically. The concentration that we used for the membrane comes from the some rats experiments, because we need accurate membrane composition. But anyway, independent of that, what you see here is that the first one is, so that we have circumcoronan here and dodecaine here. For example, crossing the rat, the Golgi here, okay? And so those two are dodecaine and circumcoronan, and the one here is vancomycin. So no matter where in this red box that is corresponded to the cell membrane, those two hydrocarbons, dodecainate, will jump in much faster than even an antibiotics, vancomycin, okay? So thinking about how those species interact with the membranes, with the biological system is important. And I know it's a stretch, but you have a bigger picture. You go from a fuel that you are designing, you can think about an emission, and then you can link to some health implication, okay? It's more complicated because, of course, when you translocate in the body, there is a lot of interaction that you have. But in principle, if I do that with the pure dodecaine or circumcoronan, I get a time of entry, a tau, that is much shorter than the vancomycin. Dodecaine actually is the most permeable compound. So the membrane, if you have a shorter and saturated aliphatic chain, they have order of magnitude higher times of entry. So if you have saturated, sorry, I want to show you again. Here, if you have this one, it's much easier for them to enter in the membrane rather than something that is not flexible, and so it doesn't allow for to move, for the molecules to travel. So the bottom line is that, again, this is a blow up. It will be much easier for you to see. Sorry about that. But the idea is that, again, time of entry, different membrane, dodecaine and the circumcoronan, they have a much shorter time of entry. Okay, that was something that I got. Okay, so I'm going to stop here. We'll take a last break, and then I'll show you one more application, and then we're done.